Otezla is the first and only oral treatment approved for adults with plaque psoriasis across all severities and the only approved treatment for oral ulcers associated with Behçet's disease. It is also approved for adults with active psoriatic arthritis. Tonight we honor Han-Hua Man, George Mueller, and Peter Schaefer. This group of researchers from Amgen and Bristol Myers Squibb are recognized for Otezla, apremilast, an oral medication approved by the U.S. Food and Drug Administration in 2014 for the treatment of certain inflammatory conditions, including moderate to severe plaque psoriasis and active psoriatic arthritis. It was also approved in 2019 for the treatment of oral ulcers associated with Behçet's disease. Otezla was also approved in 2024 for the treatment of moderate to severe plaque psoriasis in children and adolescents ages 6 or older. Otezla works by inhibiting phosphodiesterase 4, PDE4. PDE4 is an enzyme that plays a key role in the regulation of intracellular levels of cyclic adenosine monophosphate, CAMP, a messenger molecule involved in various cellular processes. PDE4 modulates inflammatory responses and immune cell activity by breaking down CAMP. Inhibitors of PDE4 help to decrease the production of pro-inflammatory cytokines and other mediators, alleviating symptoms associated with these diseases by preventing the degradation of CAMP. Otezla, an inhibitor of PDE4, is used therapeutically to reduce inflammation and treat conditions like psoriasis, psoriatic arthritis, and Behçet's disease. As an oral medication, Otezla offers a convenient alternative to injectable biologics, making it a valuable option in the management of chronic inflammatory conditions. Since its initial FDA approval in 2014, Otezla has been prescribed to more than 1 million patients worldwide. In the 1990s, Dr. George Mueller and Dr. Han-Hua Man from Celgene, now Bristol Myers Squibb, were focused on making small molecule inhibitors of TNF-alpha production. Dr. George Mueller led the discovery project team, modifying the chemical structure of thalidomide compounds to dial out unwanted side effects and improve the anti-inflammatory properties. In 1999, Dr. Han-Hua Man first synthesized the PDE4 inhibitor that was later named apremilast. This compound had many potentially useful properties, including inhibition of TNF-alpha, IFN-alpha, and IL-17 production, and had the potential to be developed into an anti-inflammatory medicine. Dr. Peter Schaefer was responsible for the biological testing included in the IND and NDA applications, which supported apremilast's development. Early clinical biomarker studies using blood from healthy volunteers, as well as skin lesions from psoriasis patients, demonstrated the pharmacodynamic inhibition of TNF-alpha by apremilast. Further clinical results in plaque psoriasis, psoriatic arthritis, and Behçet's disease resulted in FDA and EMA approvals of apremilast, commercially known as Otezla. The American Chemical Society recognizes these heroes of chemistry from Amgen and Bristol Myers Squibb for their outstanding contributions to science and their service to society.